Good evening, everybody, and welcome, and thank you so much for coming here tonight for this public lecture. My name's Trina Burgess. I'm the director of the Harry Butler Institute here at Murdoch University, and we're really glad to be hosting Shannon during her time in Perth in this lecture tonight. So first of all, I'd just like to acknowledge that Murdoch University is on the lands of the Whadjuk Noongar people, and I would like to pay my respects to elders current, present and emerging, past, present and emerging. And this has been lands where learning has been undertaken for millennia. We've got a wetlands here, beautiful wetlands down the back of the university, and this has been a really special place for a really long time, and we're really glad to continue on in that tradition. So, what am I going to say about Shannon? Not very much as an introduction, because she's going to tell us a little bit about her pathway into this topic during the beginning of her talk. But I just want to say that she is a forest pathologist and a tree health expert in the United States in California. So this organism that we're talking about, this very topical organism, Polyphagous shothole borer, belongs in a group of ambrosia beetles and bark beetles, many of which carry fungi with them, and these fungi cause tree diseases. Of course, you're going to hear a lot more about that, but one of the really big things is in the Northern Hemisphere, these are quite common, and there are quite a few epidemics that have been caused by ambrosia beetles and bark beetles in the Northern Hemisphere. This is a new thing for us in Australia. We don't have any native, we have native bark beetles and we have native ambrosia beetles, but they've never caused a pandemic or an epidemic or this sort of death of trees as we are seeing here. So this is something new for us, and it's really important that we look to where this work has been done for a century or more to try to understand it a little bit better. So, during her career, Shannon's worked on many different pathosystems, including this one. So she's a very experienced forest pathologist with a really good understanding of a lot of different systems. She found Polyphagous shothole borer in California. So we're not just talking about somebody who knows something about it. We're talking about to the person who actually discovered it was there and has conducted research and work on it ever since it first arrived there. And I guess the other thing that I wanted to point out is that a lot of the decisions that are made there are based on the science. And the other thing that I want to say, because I keep on congratulating her, she's just recently been appointed in the forest pathology department at UC Davis. This is a really prestigious forest pathology department, and it's like done some of the best research, or plant pathology department, with really good forest pathologists, and they've done some of the best research in the world in forest pathology. And so, welcome, and please proceed. Thank you.
Thank you. Thank you. Thank you, Trina, very much for that very generous introduction. It's really an honor to be here, and I'm grateful to come and talk to you all today about my experience working in this system. So I'm going to talk about PSHB lessons in management and treatment from Southern California. And just to orient you on this map, this is a map here of California. And here is UC Davis, where I currently work. So UC Davis is in Northern California. And I've been involved with this issue, as Trina was saying, down here in Southern California. Let me see, oops, there we go. Okay, down here in Southern California, since we discovered the problem, me along with Dr. Akif Escalin, on a backyard avocado tree in 2012. And so I wanted to first acknowledge that this work is part of a multi-campus, multi-agency collaborative effort to develop essential building blocks for integrated pest management across the state. So it's a coordinated effort among different entities that involve agriculture, academia, city planning, the recreation, fish and wildlife, as well as regulation. And all of these different stakeholders have a vested interest in the problem. and for different reasons. And a lot of the work that we've done over the years, both formally and informally, has been an ongoing and collaborative effort to address this issue. So recognizing all of that for my talk today, I'm going to first introduce the pest pathogen system in California and kind of put it more into a global context. I'm going to then launch into the research framework that we've implemented and discuss how that fed into a statewide initiative to respond to the problem. I'm going to explain what we know about management options in California based on what has been tested and based on the research that we've done. And then I'm going to end with a case study, some of the management lessons that we've done using a case study that we've conducted in Disneyland, because why not finish with the happiest place on earth, right? So Fusarium, so introducing the pest-pathogen system. So Fusarium dieback invasive shot-hole borers is a pest pathogen complex. It's caused by the combined effects of these specific Fusarium fungal species that are vectored by these Euwallacea ambrosia beetle species, which together are commonly known as the invasive shot-hole borers in the United States. So we have Polyphagous shot-hole borer. We have two beetles in California. We have the Polyphagous shot-hole borer and we have the Kuroshio shot-hole borer. They're morphologically indistinguishable, nearly indistinguishable, but genetically distinct cousins to one another. So just to simplify things, we refer to them as invasive shot-hole borers in California. And so just to back up a little bit, what are ambrosia beetles? So ambrosia beetles are this huge diverse group of wood-boring insects that have these special pockets in their body called mycangia. And they use those mycangia to carry fungi. So depending on which group they belong to, those mycangia, which are shown in the body on these different beetles here, this diverse group of beetles here in the yellow, depending on what group they belong to, they're going to be on different parts of the body. And so this is a typical family photo of the invasive shot-hole borers within the host tree here. And basically they share this ecological strategy with thousands of these other species of wood-boring insects, where the mature female bores into the wood of the host tree and inoculates the gallery wall with the symbiotic fungi that's carried inside of her mycangia, which for these species of beetles are located in the mouth. They bore into the trees, but they don't consume the tree tissue. Instead, the beetles depend on the fungi as a source for the larvae and the rest of the family, and the fungus depends on the beetle for dispersal to trees. So and the rest of the family here. So the beetles are vectors because they pack the fungal spores into their mouth parts, those mycangia, to carry the fungus from tree to tree for their family, basically. And so this is a simplified life cycle of Fusarium dieback, and I simplify this because it really depends on the host and how these things behave from tree to tree. But just to give you an idea and a picture, unlike the majority of most ambrosia beetle species, these attack apparently healthy trees. The female basically bores into the tree. And when she bores into the tree, she's going into the wood, and she basically makes a gallery into the wood, which is basically a tunnel, and she inoculates that tunnel with the fungus where she lays her eggs, and then that fungus is also the food source for the larvae and the developing beetles inside. At the same time, the brothers inside there, they mate with their sisters. This eliminates the risk for needing to look for opposite side of the sex outside of the tree. That's actually really important. For this reason, this class of ambrosia beetle doesn't need to produce sex pheromones. That has important management applications. So they prefer to produce a new gallery on the same tree near the mother's gallery. So the young female adults, they'll leave that gallery and they'll walk, literally they'll walk outside of the gallery and they'll just go down a couple centimeters and they'll make their own gallery right next door to their mother. It's a risky business to fly for these beetles, so they're gonna stay on that same tree as long as they possibly can. At the same time, the fungus is a pathogen, so the fungus is colonizing onto that tree. And what happens is as the fungus colonizes into the wood tissue, the host basically gets scared, and it produces what's called tyloses. So these tyloses plug up the xylem tissues, which are responsible for taking up water and nutrients into the plant. And that essentially, it's an overreaction by the host because it's trying to prevent the fungus from moving systemically through the tree. And so for that reason, what happens is it's essentially choking itself. That causes wilt and branch dieback, and on some trees, it kills the plant. So then when the host can no longer support reproduction of the fungus itself to support the reproduction of the beetles, or when the host dies, that is when finally the beetle leaves the tree and goes and tries to find a new host that's healthy, that can support the fungus to grow inside those galleries. Okay, so the story becomes a little bit more interesting. So the beetles do carry other non-fusarium fungi in their mycangia. Each of them play a different role, we think, in terms of their development. But the fusarium is the essential and dominant fungus. I just wanted to mention this because it is important to introduce some nuance here. Okay, so here is a global distribution of all of the Euwalacia species where they have been introduced. Here is the native range of Euwalacia. So there's a whole bunch of Euwalacia species, and they are native to Southeast Asia. Not all of them are destructive or attack living and healthy trees like PSHB and KSHB. But all Euwalacia species carry these special ambrosia fusarium fungi, which happen to be part of this special group called the ambrosia fusarium clade. So we call that AF. Here is the global distribution of PSHB, where it's currently in Israel, California, South Africa, and Western Australia. There's actually a cryptic species of PSHB that's recently been detected in Argentina as well. We originally thought that there was this one-to-one relationship between the beetle species and the fungal species that it carries. So for example, the PSHB carries only Fusarium euwalacea, but we now understand through some research and obviously through what the beetle carries in Western Australia, that this relationship with ambrosia fusaria group is non-exclusive because the PSHB in Western Australia carries a different ambrosia fusarium. So just to kind of sum that up, in the PSHB in Israel, California, and South Africa has been introduced on separate occasions, but it carries Fusarium euwalacea. In Western Australia, you get this other Fusarium AF18, which that's what you have. Okay, so back to California. So this is the range where we have confirmed Fusarium dieback attacking trees in Southern California. The beetles were introduced to Southern California on 2 separate occasions from 2 separate locations in Southeast Asia, presumably on packing material. But the story goes back a bit earlier than that. So for PSHB in California, a single beetle was first detected in a state monitoring trap in 2003. The problem or the damage, however, wasn't recognized until 2012 when it was discovered on a backyard avocado tree in Los Angeles. And then a second introduction, which was the KSHB in late 2013, occurred in San Diego County. So that's in the blue. That was in the blue in the previous map, was detected in commercial avocado groves. And then just this last November in 2023, nearly a decade since kind of like its northernmost detection, PSHB was confirmed on infested sycamore trees. So this is 280 miles north in San Jose, 280 miles north of the leading edge of the infestation. And so this has actually set off some major alarm bells in California, because now it's in Northern California. This is a major leap for us in California. Okay, so very early on, we observed that there was a whole range of symptoms on a variety of tree species. So this is just kind of a few examples of some of the kinds of different symptoms we would see on a bunch of different tree species. This is not all inclusive. And the complex also attacks many tree species that are distributed across many different kinds of systems, including native plant communities, urban forests, as well as avocado groves. And so originally, early on, California produces 90% of the US domestic crop, so this was a major concern as well. And depending on the host and the location, the impacts can be really devastating. So it is something that we took very seriously. So for example, this is looking over the Tijuana River Valley in Southern California, right near the Mexican border, near San Diego. And within 1 year, over 200,000 trees were killed in this riparian forest. So this is kind of like the worst case scenario where we saw this mortality. And we saw cascading effects of that mortality as well. So looking kind of standing on the ground there, this is at that same location where you see that mortality. We saw 9 months later castor oil, that highly invasive weedy plant, began to encroach into the area, preventing establishment and recovery of our native willow species. And those native willow species serve as critical breeding habitat for endangered bird species. And so there was these cascading effects of that mortality. So because this is a multi-land use and multi-stakeholder issue, we quickly realized that we needed to develop a framework that addresses the complexity of this issue, so that we could take action using whatever knowledge is available, at any given time. So our approach was to develop an integrated pest management strategy as part of our research program. And the essential IPM components that we identified include monitoring and developing tools to quickly identify the disease-causing organisms and detect new infestations, researching their basic biology and life history, conducting risk assessments to identify which tree populations and forest communities are most vulnerable to the establishment and most important in their spread, developing and evaluating biological, mechanical, and chemical control options that are tailored to the appropriate needs for particular settings, and then sharing the menu of options and improving IPM strategies with stakeholders in an iterative process. So none of this was all kind of not linear. This was all of these essential building blocks were kind of happening often at the same time and getting revised over and over again as we were learning new information. And this all evolved into a statewide initiative through the California Invasive Species Advisory Committee, which is basically a body that advises the State Interagency Council for invasive species on best measures to mitigate ecological and economic harm that's caused by invasive species. And in 2019, we developed a statewide plan to respond to ISHB and Fusarium dieback, and I was in a leadership role in that whole process. If you want more details, you can refer to the report. I put a link to it up there, and so this is being recorded if you want to take a picture. But briefly, the SISAC committee directed the development of the plan that addressed 4 key elements of the plan and corresponding subcommittees. So those were green waste and firewood as pathways for movement, survey detection and rapid response, research and technology development, and outreach and education. And so basically the essential elements of the plan required consensus building between these different stakeholder groups and organizations to develop priorities on how to allocate funds across these plan elements with the overall goal of controlling the pest pathogen complex on a statewide level. And then this whole process actually turned into a sociological case study on collaborative governance that I wrote about that's out for peer review right now. So that should be out, looking forward to that coming out. So I'm not gonna get into all of the details here, but basically the consensus out of that process was to fund a bunch of research projects that we all came to a consensus on. We hired a statewide trapping coordinator, statewide communications coordinator, and there were some key partnerships that got formalized to help mitigate that otherwise could not have come together to help mitigate the movement of green waste and firewood as pathways. So coming back to management options in California, I want to start by talking about the host range first, because that's going to be the critical piece when we talk about management. And that's because we observed a lot of variation very early on in not only in the broadness of the host range, but the severity of the impact, where some hosts just shows branch dieback when they're attacked, where others are killed when they are attacked. And in order to understand this variation across different species, we dug into the biology a little bit more. And what we found was that when the beetle tries to attack a tree, it could not establish a gallery and successfully colonize on some species, while for other, the beetles couldn't establish a gallery, but the pathogen could colonize on the tree. And then for other species, a beetle could bore into the trunk, produce a gallery, establish a fungal garden to feed on, and then make their brood, and then spread to a new host. So both the beetle and the fungus must be able to reproduce for the host to be what we call competent, or to be a what's called a reproductive host. And fortuitously, some of the most heavily infested areas in the very early stages of the infestation in Southern California were home to 2 of the biggest botanical gardens, because they harbor a wide range of species that represent unique and common ecosystems from all over the world, including those that are native to and planted in Southern California. So we had this rare opportunity to use these gardens as a natural laboratory to investigate plant host range and the beetle fungus complex. And what we found was that these host types are based on these nested outcomes of interactions between host plants, the beetles, and the fungal pathogens. And this was also integrated into the hosts. We integrated what we observed in the botanical gardens in California, as well later we integrated the information that we got from the host range in South Africa as well. And so basically the beetle makes an attempted attack on 324 species, so where it just makes an attempted attack. Only 137 the fungus can colonize, but it's 77 species which are the competent hosts that support that reproduction of the fungus and the beetle, and those are important in the spread. And so those include 20 that are native to California, which dominate the landscape in California, avocado, and anywhere from 25 to 60% of all individuals that are planted in the urban landscape in Southern California. But then a smaller subset of 18 species can be killed when they are attacked. So the lethality of different hosts is important in terms of ecological impact. We also know that if you get more into these nested outcomes, the hosts become actually more closely related to one another, which is really interesting. And that has allowed us to do some really interesting predictive modeling where the pest-pathogen complex is likely to establish and cause the most damage across the state. I don't have time to go into the details on how we developed that model, but this is basically the risk map that we developed based on our understanding of host relationships, as well as climate impacts on the beetle, and has been able to basically inform, strategically where to do certain, to strategize decision making, and where to, use the most, our limited funds for management, where to allocate those strategically. But in terms of management in an urban forest, where we kind of have those things best worked out based on the research that we've done, an understanding of host range and its variation and susceptibility among hosts is important because it means there's no one-size-fits-all approach to management of this problem because of the complexity of the system. So what we did was we developed this management matrix to guide which actions to take depending on the host type. So whether or not it is a reproductive host, as well as these other factors, so including the infestation level. So if it's not infested versus low, moderate, severe, or low, moderate, heavy, or severe. The tree value, so whether it's a species of high economic or cultural value, like a heritage tree or a larger tree, versus a tree that's low value, so something that's smaller, has structural problems, that is like a poor economic value. And then the hazard level, so something that poses a risk to people and property versus not. And I'm not putting this for you to read the fine print, but just to highlight the complexity of what management looks like in this system. And again, we have our host types. Here, we have our hazard level, we have the infestation level, and then we have whether it's a low value versus high level tree. And then in the white, those are the actions that we want to take depending on those different factors. And the main thing I want to point out is that pesticide use is reserved for high value trees only. It is a last resort. It is not a magic bullet. There's no magic bullet in the system. Only severely infested trees or hazard trees are removed. And monitoring and branch removal to reduce the beetle population is the most common response. If you don't like a matrix, there's a flow chart. We're trying to get at all learning styles here. So these are all available on the website. It gets you to the same place. Okay, so what does monitoring look like? Okay, so monitoring for ISHB, there's visual inspections, and you can monitor through trapping, and they are complementary to one another. So monitoring involves looking for signs and symptoms of the disease. The beetle, this is actually on the left. This is actually the beetle sticking with its abdomen sticking out of the entry hole of its gallery. So it has a very interesting behavior where once they establish a gallery, they sit at the entrance of the gallery and they guard it. And that is because that's a sign that they have gone in, they've laid their eggs, they've inoculated the gallery with their fungus, and they have an active gallery, and there's a family in there. And they go and they stick their abdomen out to guard the gallery from any invaders getting in. So oftentimes you'll see like a little beetle behind sticking out of an entry hole. Those entry holes, we're lucky in California, we don't have anything else that's the size, roughly 0.85 millimeters, and they're perfectly round and regular, and they're roughly the size of a tip of a ballpoint pen. And so that is something that we look for. The symptoms depend on the host itself. And all of the different pictures that you see here between staining, gumming, this white powdery exudate, that's a sugar that's produced by avocado, or frass, those are the response that's specific to that particular host. So it's basically the host's way of trying to respond to try to push the beetle out of the gallery, or out of it, trying to make its gallery and protect itself. So those are the different kinds of symptoms that you'll see. Visual inspections in California, they tend to be more prominent in the north and eastern sides of the trunk, main trunk and branches. And typically during monitoring, we assign to an infestation level. So severely infested trees have more than 150 holes and where dieback is observed, versus heavily infested trees have the same amount of holes, but we don't see dieback. And so that's an important nuance to understand. Monitoring for beetle activity, so monitoring whether the holes are active or not, can inform, determine how the tree is responding, and can help inform maybe which actions to take as well. So active holes, the hole's well defined, you see the host is responding in a way, so you might see the staining and the weeping, or in an avocado you might see sugar coming out, or frass, the beetle is sticking out, versus an inactive hole. There's none of those, the beetle's not there, the host isn't responding. And sometimes you'll see what's called compartmentalization. That compartmentalization is actually the host responding by producing what's called a callus. It's creating a wall, and it's closing the entry hole. And that's actually a sign that the tree is doing a good job of recovering from the beetle attack. And so that is a hole that is no longer active anymore. And so one of the things that we want, that's important to understand is if a tree is on its way to recovery, there may not be any need to remove a tree or to cut it down. One way to confirm whether or not there is beetle activity is we have this little trick. So when you plug that entry hole with anything, so if you plug it with tape or plug it, with anything, the beetle can't stand it because it needs that hole to be open for oxygen exchanged in the hole itself. And so we've taken, as soon as you unplug it, the beetle will come and immediately try to unplug that hole. So we have taken advantage of this behavior and use a water-based latex paint and paint the entry holes of these holes to see whether or not those are active or not. And you can see that the beetle within seconds will come and immediately open up the hole. If it stays and remains clogged, then you know that that gallery is no longer active. And that's actually a really great way to test if your treatment has been effective or not. Okay, so trapping is another monitoring tool that we use. The uses for trapping include early detection, guiding inspection efforts, and trying to identify the best timing for treatments. There's a whole range of different traps that you can use. We tend to prefer the medium to large size bark beetle traps, those sticky traps that get folded in half. There is a special kind of lure that contains querceverol that is based on the scent of the fungus itself that attracts the beetle that lasts 1 to 2 months. So we use that lure. The traps, what we've learned is the most effective is to place them, at least 50 meters apart. And I say this, it might sound obvious, but the important thing that we've learned in our lessons from trapping is not to hang these traps on branches. And I've seen this enough times that I need to say it, because hanging the trap on a branch actually turns the tree into bait, right? Because you're essentially attracting the beetle to the tree, and you're turning the tree into a trap. And then the tree becomes like heavily infested. We don't want that. We want to use the trap as a tool to see if the beetle is in the area. And then if we see the beetles in the traps, then we do ground truthing and inspect the trees in the surrounding area. So we recommend using a pole, placing the trap roughly 0.7 meters high on the pole, and trapping that way. And then this is just, again, to continue with trapping, this is a graph that is showing the nuance between different sites, a couple of graphs showing the nuance between different sites. So this is showing the proportion of beetles that are caught over time at 2 different sites that are within the same county in Southern California. And typically you get a peak in the spring and the fall when the beetles are flying and when we catch them. But as you can see that there is some nuance there and there is some variation between sites. And so this is where trapping can be a really useful tool for very specific sites on on how to monitor the beetles so that you can be strategic on when to apply different treatments. Okay, so removing infested branches so there's monitoring, and then there is using that monitoring in combination with doing sanitation practices. Those are kind of the most common things that you will see in that management matrix. And removing infested branches is probably the most, is one of the most common things that we do. Oftentimes, there's not really a need that we have found to remove a whole tree, because what we have done is we have basically managed to, if we can, our objective is to reduce the population of the beetles in that particular area. So this is especially a good option for some tree species where the tree doesn't even attack the main trunk. And so, so removing branches is one of the most important things that you can do. And again, just to show why it's key to not just kind of go towards removing a whole tree first, and why kind of reducing the population and removing infested branches is a useful, that we found has been a useful tactic, is because we have seen recovery on trees when we have done this. And so again, this is just showing a few examples where we've seen that compartmentalization on trees that have been attacked, and that they've managed to recover after being attacked. And so what we recommend, or what's been recommended in the management matrix, is the severely infested trees that have more than 150 holes and that exhibit dieback. So after you've done your monitoring, after you've done, branch removal, maybe it's a heritage tree, and you've done some of the chemical treatments that have been applied, and the tree just is failing, or it's just so far gone that it just needs to get removed. Then if it's just too far gone, removal ends up being the only option. That's that has to happen. But again, it's typically reserved as one of the last resorts. If tree removal is an option, then it's really important not to just remove the tree, but to remove the stump as well. We've seen in a lot of cases that the tree gets cut and the stump gets left behind. But what happens is the stump is harboring lots of beetles, and we've seen this over and over again. So one of the big things, one of the big lessons that we've learned is that after a stump has been cut, all of these beetles that are still in the stump, what happens is, again, the beetle needs healthy trees to reproduce and survive, right? So as soon as you cut the tree, the tree can no longer support reproduction of the fungus and of the beetle. The beetle is going to freak out, and it is going to do a mass dispersal out of those stumps, and it's going to fly somewhere else, and it's going to start attacking other trees. So it's really, really critical to treat the stump by using a stump grinder. So not only do you need to remove the tree, but it's really critical to remove the stump. And how do we manage infested material? So chipping, the data has been, the data show, and the studies have shown that chipping to less than an inch, or as small as you can, but chipping to less than an inch, the studies have shown that kills 99% of the beetles. You can then use those chips locally as mulch in the same property. If you want pest-free mulch, if you just want to be really have a clear head about it, you can chip and then compost, and that would kill everything. If chipping is not an option, then the logs can be solarized with a clear tarp, and, or they can be kiln dried. Okay, so as far as chemical management is concerned, one thing that I want to kind of mention first is that the research is based on California conditions. So this is based on research that we've done in California based on California like constraints, regulatory, and environmental. So just kind of want to mention that, first. And I also want to start by saying that research shows that none of the chemical treatments work 100%. The first thing also the next thing I want to say is only consider treating infested high-value trees. So in that management matrix, this was only reserved for a very small subset of trees. Those trees that are high value, high cultural, important trees. This is not a preventative treatment. There are no preventative treatments. This cannot be applied as a preventative treatment. It's generally reserved for moderately to heavily infested trees in an urban forest. A combination of insecticide and fungicides is recommended. You have to consider the tree health, the location, the tree value, and the level of the infestation before applying these treatments. It's only available to state licensed and county registered pesticide applicators. And again, none of these treatments work 100%. So with all of that being said, these are what we know have been effective based on what the research says. So for trunk sprays, the trunk spray that is available, and this is appropriate on heavily infested trees or locally after you remove an infested branch. There's a chemical called bifenthrin, which is a contact insecticide, plus Bacillus subtilis, which is, it's a biofungicide, it's basically a bacteria that is an antibiotic that's effective against the pathogen. It actually kills the pathogen. And the goal is to kill the beetles that are about to disperse or that landed on the tree looking for a new host. It should be applied when the beetles are dispersing, typically early spring or late fall. So that's where the trapping data can be useful. You can spray trunk and big branches, avoid flowering plants and areas close to water. And then the other option is systemic pesticides. These are taken up through the tree, so killing the insects and fungi inside the wood. This depends on the tree uptake. So these, in our case, we apply them in the spring or the fall. The trees need to be irrigated before and after treatment. The options are a soil injection with imidacloprid, which is an insecticide. Imidacloprid is good for sap-sucking insects, but PSHB doesn't eat sap. So this is not like, but it's a very highly effective, it's highly systemic. And so that's one of the reasons why they use it. And so there's some studies that say that it's been somewhat effective, but what is more effective at killing the beetle is through the trunk, is emamectin benzoate, but that can only be applied through the trunk injection. And that's an insecticide that's applied with propiconazole, which is a fungicide. The fungicide is locally systemic. So here's the thing. The trade-off is emamectin is not as systemic as the imidacloprid. So that's the trade-off there. And the propiconazole is only locally systemic. They've seen in some studies with other beetle pathogen systems, like in laurel wilt, that it can be effective at killing the fungus. However, you have to keep applying it over and over again because it moves very slowly through the plant. These can not be applied on heavily infested trees because the tree itself is already compromised so heavily by the xylem tissues, right, that are taking up the water and nutrients are compromised by the galleries that are being produced by the beetle and all of the tyloses and the fungal colonizations that are being produced by the fungus. And so because of that, this really isn't appropriate for heavily infested trees. So, if it is going to be used, it's going to be used on moderately infested trees if you're going to use it. It does require drilling into the tree, and one of the other trade-offs to that is that you can create secondary infections by opportunistic pathogens. So you're creating a hole in the tree. That's a nice big infection court for other canker pathogens to come in that actually move much more quickly than the Fusarium pathogen does and cause even more damage, especially if you have to drill over and over and over to apply these pesticides. So just even once we get to our last resort, treating these trees, the decision on what treatment to use depends on the tree health and the location. The main message here is don't just apply a pesticide just because you have an infested tree. The goal is to use these other tools in the toolbox, which is why we really encourage monitoring and removing infested branches in California. That has been the most effective approach at reducing the beetle populations in the urban forest, and then using these other treatments as a last resort. So I'll end with this little case study in Disneyland, because we had this opportunity to work in this really extremely diverse urban landscape. That's this unique and interesting model system. You wouldn't think that Disneyland was like this kind of, it seems odd, but it seems odd. But the resort, it occupies about 600 acres. It has theme parks, hotels, shops, and parking lots. But there's also approximately 16,000 trees in the park and 681 species and varieties. So it's actually a really diverse hotspot. And it's a really diverse urban forest. And it's also in the middle of where shot hole borer was, it's in the middle of like the invasion. And so early on, when they had the initial invasion, they decided that they were going to be proactive, and they picked 600 non-infested trees and they injected them all with systemic pesticides. And they kind of panicked. And as a result, they got this instead. And so a lot of their trees got this kind of damage, where they got huge cankers on their trees. So instead of getting shot hole borer, they got canker pathogens on 600 trees. And these secondary infections, like I said, move so quickly through that it just burned through a lot of their trees. And so that's how we got involved with the problem. So we got a team together. Dr. Escalin, John Kabushima, Rhonda, the urban forester at Disneyland, as well as Heather Knoll. She's the IPM manager. And we put this team together to implement an IPM program there. And so basically, here is Disneyland Resort on the right here. Here's the park. And what we did was we placed traps around the perimeter of the park just to see, just to monitor anything that might be coming into the park. And then we also placed traps on the interior of the park so that we could identify any hot spots of where the beetle might be occurring inside. And then once we identified hot spots, the crew, we trained the crews, the horticulture department there, to monitor inventory trees to determine the source of beetle populations on those trees and to determine which management actions to apply based on attack severity and host type. So we did these monthly visits to evaluate the findings and decisions, and then to make on findings and decisions on decision making for what next steps were. And the management actions we had available to us, again, were cultural sanitation, so pruning infested branches, applying a pruning wound protection, which is basically a commercially available version of Bacillus subtilis, or cease, so that antibiotic to prevent any canker pathogens from entering pruning wounds, and if necessary, removal of heavily infested trees, and if necessary, trunk and or injection of systemic pesticides if necessary, and trunk sprays. But this also involved a lot of working with the staff in an iterative process and sharing the menu of options and improving our IPM strategies on and on and on as we were kind of going through this process, as they were learning how to identify signs and symptoms of the beetle, and they were learning how to manage these trees in the park. And so so just for an example, and so, just an example of some of the things that we did, and just as an example of a success story. So, here's an infested valley oak that had hits on the branch, and so we decided to remove that branch from the tree and continue to monitor that tree month to month. The tree still had hits on it, and so basically after doing a local spray of bifenthrin, so this is just on a very local scale, like underneath the branch collar, not doing like a whole full scale, like shh, but being very strategic about where we're applying this. Spraying bifenthrin, putting it right at that location, the tree managed to recover. And so by doing this, by removing the branch, we reduced the beetle population on that tree, reduced the ability for the beetle to, the population to spread to other trees, and then we protected that tree and allowed that tree to recover. Early detection and monitoring also allowed us to find hidden sources of beetles in tree stumps that were harboring beetles throughout the park. Old stumps of things, because the population pressure was so high, there were old stumps of bird of paradise that aren't hosts that had beetles on them, that just blew everybody's mind. But it was because of these active monitoring efforts that were going on in the park, we were able to catch these things early and remove them and do something about them. And so after all of these interventions, less than 1% of the trees were actually chemically treated with injections in the park. Just by doing good sanitation practices. And really all we did was we did an IPM strategy. We identified the pest and pathogen accurately by training people. We monitored and assessed the disease throughout the park actively, established guidelines for when management action was needed, and used a combination of cultural and chemical management tools, and did a lot of training and outreach in the park. This is a graph that's showing the number of beetles that were caught in our traps over time. And so the population at the beginning was really, really high, and over time, it dramatically reduced. We dramatically reduced the beetle population in the park through, because we were monitoring the effect of this program. And so, Disneyland is still living with the beetle, but the population has gone down so much that it's no longer a huge problem and causing massive dieback on their trees in the park. So with that, I'm going to skip that, and I'm just going to basically end with this. So shot hole borer in California is something to be taken very seriously. But we have come to terms with the fact that in California it is here to stay. And so, we don't see the need to panic. It is manageable with timely action. We tell people to educate themselves about what shot hole borer looks like and other pests and diseases, get educated on what it looks like and what lookalikes look like, to take preventative actions to avoid problems, look for science-based management options, and use other tools in the toolbox first and leave chemical management as a last resort because it doesn't work 100%. And that is all I've got. So I'm happy to take any questions. Thank you.
Well, Shannon, thank you very much. I think we're going to have a few questions in the room, probably a lot of questions in the room, so we'll just see how we go. So, first questions. I think Amy here's got a microphone.
Thank you very much for that. Appreciate your time. Question, once you remove the branches, what do you actually, do they get treated, or what do you do with them so it doesn't, what happens to the branch?
What happens to the branch when you remove it?
Correct. Normally you just put it through a cutter. I'm not an agriculturist, so I don't know. You normally put it through a cutter and it just shreds it all. But obviously, if it's full of beetles or whatever, how do you manage it so that they don't escape or anything?
As soon as you remove the branch,
Thank you.
You have to chip it. Yeah, chip it right away. Yeah. No, chip it on site.
Thank you. Excellent presentation. I greatly appreciated it. Just wanted to know, in 2012 when you found it, was there any attempt to eradicate it?
There was not an attempt to eradicate it at the time because we were just trying to get our heads around the problem. This was essentially in California and in Israel. We were both kind of dealing with the problem at the same time. And we were just discovering what this thing was and trying to understand what it was. We thought it was T-shot hole borer. So we were just discovering and trying to understand what this was. And so the government didn't have as much of a grasp on how to deal with it you know, as we did, as you guys might, yeah.
Shannon, that was great, thank you. In your little charts of different boxes going down, there was, I think, about 70 species where post-infestation you have the fusarium persisting but not the beetle. Was there any phylogenetic similarity between those host trees or any commonalities in traits for those 70 that the beetle wasn't able to persist even though the fungus could?
Yes, so yeah, great question. So basically in that box, those nested outcomes, yes. So there is a phylogenetic signal in that host range, and that phylogenetic signal becomes more pronounced. So basically the hosts, you know how I was saying the hosts are more closely related to each other in those like 18 species that are attacked. Those are really, really closely related tree species. But so it becomes more pronounced as you get more and more down into those nested outcomes. So the ones that are fusarium colonized are more closely related than the ones that are just attacked and nothing happens at all.
With the ones, let me add to that, with the ones that are the fusarium colonized, will the fusarium kill the tree on its own?
No.
Okay, so it needs the beetle to keep on like spreading it through because
Yeah.
it's a weak pathogen.
Exactly, yeah.
Yeah. Do you wanna go that one?
Thanks, Sharon, very much for your presentation. I'm just on the issue of eradication. I think is it San Jose in Northern California where it's recently been discovered? That outbreak was only discovered last year, I believe, or earlier this year. I'm just wondering about the nature of that, when it was discovered, how extensive it was, how long you think it might have been there, and also, presumably at that time, you knew that there was an eradication response happening in WA, but yet I understand that you still chose not to try and eradicate. And I'm just wondering about the thinking behind that.
So there's been a lot of a huge coordinated effort to try and get rid of, to try and do something there. The hang up with that is we have multiple land ownerships in that region, and so there's the decision on who gets to take those trees out was not quite clear, and so that's been a difficult. It's been a little bit difficult.
You're being very, very careful.
Yeah.
They're working on it. They're actually working right now on getting funding with the California Department of Food and Agriculture to actually do a massive removal of trees in that area.
Thanks so much. That was an awesome talk. So can you burn the removed branches instead of chipping?
Conceivably you could. The issue with that is we have a lot of fires in California, and that would.
So it hasn't really been tried?
That would not be recommended. Yeah.
So it hasn't been tried, or?
It hasn't been tried because it would be a hazard.
Yeah.
Yeah.
Okay. Yeah.
Thank you.
Yeah.
Thank you so much for that presentation. It was really fascinating. I think there's been a fair amount of angst and concern here about the eradication policy that's been adopted, and at what point you decide the horse has bolted and you actually have to go towards management, particularly concerns around loss of tree canopy and impacts on biodiversity. So it's particularly fascinating to hear the results that you've had around trees that seem to recover when branches are removed and sort of compartmentalise. And I guess I have two questions. One is, have you found, because I guess one of the messages that you, from this presentation as well, is in many ways it was good that you just didn't go out and take a whole lot of trees, because you wouldn't have known necessarily that some of these trees could recover in that way. And so have you found similar things elsewhere around the world, in other places where PSHB has been, impacted other countries, that they've had similar sort of differences in terms of host response and different tree species and the way they've responded? And then also with those trees where there have been approaches like removal of branches and they've compartmentalised and seem to sort of recover, is when you monitor those trees, does that actually stay over the long term, and that you find that they are relatively free of beetle infestation for the long term?
So I would say that, I mean, as far as what's happening in other countries, I would probably defer to those experts. I can only speak to kind of the data that we've seen in California and what we've observed and what we've done. But as far as the reason why what we've seen, because we with the cultural and sanitation practices, the reason why it's effective is if you're reducing the beetle population, the reason why the tree can hold on better now is because it's not getting bombarded as much with beetle pressure anymore. So it still might get hits here and there, but if you're actively monitoring and doing some active sanitation practices, then you're constantly keeping the beetle population down, then you're giving the tree an opportunity to, recover.
Following on from that question, actually, there's a lot of comprehensive species host lists globally. Is California developing a species list of trees that can recover and compartmentalize? Is that certain species can do that, or is it sort of generally all trees? It just depends on how hard they're hit or how they're treated. Are you seeing that certain species can recover, and is there a host list being developed for that?
I can say that one certain species cannot, and that's box elder.
Yeah.
But as far as doing an extensive exploration on, the host, looking at recovery, we haven't done that particular data collection. Mm-hmm. Yeah.
Thank you,
Good
Shannon,
question.
for a great presentation. I have two questions. One is, you're talking about removing branches, but you have live infestation. You've shown a slide where you take a latex paint just to see whether it's actually an active hole or not. Why would you not paint the infected branches with something that they can't poke through, therefore containing the beetles within the wood you're removing, and therefore preventing that from spreading before you then chip it or contain it or destroy it in some way? And the second question is there a stratification of beetle infestations over the height of a tree? Like, do they only go to a certain height? Do they go from the ground all the way up into the topmost canopy? Is there a diameter, kind of minimum diameter, that's needed for the beetle to be able to establish itself?
Yeah, I'll answer your second question first. So the beetle can basically attack from the main trunk all the way up to all the way up through the canopy. It tends to prefer the larger substrate because it wants as much wood as possible to produce like a nice gallery. In California, there are two host species where we've seen kind of exceptions where it will tend to prefer the secondary, like the primary and secondary branches over the main trunk, and that's our coast live oak and avocado. And so we'll see hits on the trunk, but it's very rare. We will see hits on small diameter, but that is things like this small, but that is in places that are really, really heavily infested. It's not going to prefer to hit a really small twig. It wants large substrate. So usually it's in a really heavily infested site where the beetle is going to go in and it's going to, it's run out of space, and its only option is maybe those smaller diameter stems because there's so much competition. So to answer your first question about painting over entry holes, so the tricky thing with that is that the beetle is really effective at chewing through things. So it can chew through plastic, in fact, we don't even recommend those plastic Eppendorf tubes to carry the beetle because it can chew through that. So, so it's so you need to carry them in glass vials. So finding something that you can actually paint that's impenetrable is really tough.
I think just a little bit more on that,
Yeah.
with the nursery industry, do you tend to find infestations in nurseries?
That's a good question. Not that they tend to be too green. Yeah.
Not enough wood.
Not enough wood.
Yeah.
Yeah.
Maybe when people are moving the great big plants around or something, but
Yeah,
not normally.
You mentioned using bifenthrin with the fungicide
Yes.
for spraying. Just wondering, in some of our experiences, not with PSHB, but with other insects, is that bifenthrin acts more like a deterrent, so it scares them away rather than kills them. Have you got any comment on that?
I cannot. I don't know. So the bifenthrin, I don't know about it working as a deterrent. I know as a contact insecticide, in our experience, it does kill the beetle. So just by making contact with it, it can actually kill it. So one thing I didn't talk about, one thing I didn't talk about was that the beetle will oftentimes come out of its gallery, usually around noon, in the middle of the day, and just kind of walk around on the tree, just take a little walkabout. We say it's like a teenager coming out at noon, and then it goes back into its gallery before it goes back into its gallery. And so sometimes if you have that contact insecticide on there, just by walking around and making contact with it can affect it. Yeah.
Thank you.
Thanks for the presentation. I was just curious about your 280-mile jump north, and I'm wondering whether you investigated that. It seems like unlikely that somebody would move mulch that far. And also, I suppose another question is, has the progression been detected further south into the Mexican northern parts of the country?
Yeah, thank you for that. So has, so Mexico is, obviously the largest avocado producing region of the world, and so it's a really big concern for Mexico if PSHB were to get introduced into that country. The south of San Diego is the Big Sonoran Desert, and so there's not a large likelihood of it moving from California and spreading south. It would have to get in through ports, so from some other direction. So there's not major avocado growing areas that are close for it to get to spread there. And then, yeah, as far as, yeah, I mean, we have a statewide trapping monitoring program, and so there's virtually like nothing that's being detected between Santa Barbara or San Luis Obispo and San Jose. And so the only thing that, what we can conclude from that is that it may not be mulch, but it could be some other form of green waste that it got introduced that way, but we don't know. It could be green waste, it could be something else, but we don't know. Yeah.
Mia.
Thank you. I am really excited to read the collaborative governance paper, the case study on this. So fingers crossed for the peer review and that getting through soon. But I just wanted to touch on one of the pillars of the IPM that we haven't explored too much, which is around the community engagement and comms. And I guess just what kind of channels did you guys use, and what kind of, I guess, metrics of success were you kind of looking at in that space?
So, so metrics of success. So basically, the metric of success was the consensus on the decision-making process. So that was kind of like the big metric of success there. And then essentially just through the qualitative analysis that I did, it was really looking at kind of the value of all of kind of just like the structural systems that were in place beforehand, because we've had problems with bark beetles in the past. And so there were some coordinated efforts in the past that we could kind of build off of and work from there. And there were partnerships that were made that we could kind of leverage. So having kind of some of those existing systems in place was really important. And facilitative leadership was really important as well, having like a good, and facilitative leadership, and then multiple opportunities for new leadership within kind of the subcommittees that we had. So giving people that were in those subcommittees an opportunity to come up and take on leadership roles within those decision-making processes was really important.
Great, thank you.
Yeah.
Hello, how you going? I've got two questions. One question is, when you remove any limbs, what sanitation should you use for the tools? Like with dieback, you use metho. Is there any, because you're not going to cut the tree down and then throw out the blade, or how would you clean? Do you have to clean the equipment when?
Yeah, so we typically ethanol is our tried and true. Yeah.
Yeah?
Yeah.
Okay, sure. And then also, just with like local government, when a tree limb falls down and then like the government workers or something, they go pick it up and they go put it on their tray, and then they just they don't inspect it and then send it to the tip, and then it will get in the mulch pile or something like that. Should we like train those staff to, before it goes somewhere, have a look around the tree stump, or would that do anything, or?
So,
Like in an infected tree?
yeah, just any tree that falls over and people are just taking it away to the tip instead of looking at why.
Like,
So,
if it's an infected tree
yeah.
and they don't know, and because you said you have to dispose of it on site, because they'll go to 10 or 15 sites in one day, and then at the end of the day, then they just take it to the tip, and then they either put it in with every other green waste, and it all just disposes altogether. So I'm just wondering.
So like a tree just like falls over and you don't know why, and then they take it out and move it.
Yeah, yeah.
I see what you're saying.
Yeah.
Oh, okay. Sorry, I'm like.
I don't know.
Thank you.
I wouldn't know.
Yeah. Translate, yeah.
Translation. Yeah, usually that, like, so there's been a lot of training of like public works people to, on what shot hole borer looks like and what these things look like. So typically they know what these, what might have caused that failure. So they're not just going to take any kind of like random thing and take it away to the dump. They're going to know how to properly dispose of it. Yeah. Yeah, no, that's a great question.
Okay, sure. No, that's all good.
Yeah, thank
We're
you.
going to make this the last question unless, now there's 3
Right.
last questions.
Thank you very much.
4 last
Well-
questions. Yeah. There's, no, Ami, there's somebody here who hasn't asked a question. I think we'll leave it there after, yeah.
Hi, my name is Felicia and I actually work for the City of Swan. We are the largest local government in the metropolitan area by land. So to continue on from what that gentleman asked, there's actually a lot of education that's been provided to us within the Shire itself. We've actually had the Department of Agriculture come and visit us and educate us. Recently, in the past couple of weeks, we've actually had a meeting with our team leaders. A few of our general hands, which is just kind of their offsiders, had a meeting within the council chambers, and they actually brought in the PSHB in to show people what it actually looked like. There's a lot of imagery that's been provided to us that we know exactly what to look for. In our supervisor's office, we've got these little plastic magnifying glasses so that we can take a photo with all the measurements and stuff like that to just send it in to the relevant, I guess, departments. But yeah, we are doing what we can. We recently just had our general waste pile chipped. We had it chipped twice just to make sure that we are getting it to the size that it needs to be. Also, I guess I have 2 questions as well. When it comes to an infestation, when it first starts, how fast does that begin from obviously just the initial infestation to getting to that severe infestation that we see?
It really depends on the host and the location. So it's really variable. There's no hard and fast thing to say. It can be really, really rapid, within a few months, if it's like a box elder, or it can be not at all. So that's why monitoring is so important.
Yeah, I have actually seen myself, obviously, I work in different parts of the City of Swan. I've worked from like Beachborough, Balladura, Brabham, Dayton, so I cover quite a large area. And also, I wanted to mention, I guess that, yeah, we're doing everything that we can, really. I mean, we are only just the local government. Obviously, there is so much more that we can do. It's more about, I guess, spreading awareness about what's happening, and that's why I guess I come today to educate myself more. But yeah, that's basically it.
Okay, last question here.
Thanks. Two-part question, and the two parts are totally unrelated. So, part one, I saw a picture of the ficus amongst your species there, and I'm just wondering what ficus species you have seen it in California, and I'm wondering if you can speak a little bit to the long-term effects you've seen in ficus species in California, noting that Ficus macrophylla and Ficus rubiginosa are not on your list of 17 species, I believe, killed by the pest disease complex.
So the ficus species in California, I'll just say we don't see what you're seeing here in Western Australia. So it's not doing anything. Yeah.
That's interesting.
Yeah.
Different fungus.
Mm. And just on the bifenthrin application, it seems like you guys are using trunk sprays as the most common method of application. I'm just wondering if there's a reason behind that, or if you've tried other methods of application, like painting on with paintbrushes or anything like that.
I mean, it's a contact insecticide, so I guess just like the most efficient application is just spraying it on the trunk.
And you're doing that from the ground, obviously, a lot of the time. You're spraying it from the ground with a hose by the looks of the pictures, is that right?
Yeah.
Yeah.
Okay.
Thank you.
I think we're going to stop at 7:30, and I mean, we could be here until midnight, I think, but you're very anyone's welcome to ask any additional questions online or through any of our social media, or and we will try to get answers out on those things, I guess. Kat, what's the best way to contact us? hbi@murdoch.edu.au, and we'll get extra questions to Shannon to answer while she's here in Australia visiting us.
Thank you. Thank you.